Hello viewers, and welcome to the third part of Fabry Disease Explained. Hello Fabrienne. It's good to see you again. Today, using the terms that we explained in the last video, we will be discussing what causes Fabry disease. First of all, we wish we knew. We could tell you something like, a missing enzyme leads to accumulation of deposits in the lysosomes, which in turn causes damage in multiple organs. That's a nice story, and it's something anyone might understand. Telling it would make for a very short video, meaning Fabrienne could go and get some rest again. But Fabrienne is not the kind of woman who just tells people whatever is convenient. So we will explain what is known. And then you might not know everything about the causes of Fabry disease, but hopefully you will be able to recognize oversimplifications and misunderstandings when you encounter them. We'll start this video by diving into one of Fabrienne's cells to see what's going wrong. Fabrienne, could you come backstage for a moment? There. Now we have a microscopic slide with a drop of virtual blood. When we zoom in, we can nicely distinguish three types of blood cells. The round ones with a dip in the middle are red blood cells, those balls with a rough surface are white blood cells, and the irregularly shaped little ones between them are platelets. Red blood cells and platelets are highly specialized cells that differ in many ways from regular cells. For example, they don't even have nuclei, making them useless for our study today. White blood cells, however, have everything we need to explain Fabry disease in practice. When we cut this white blood cell in half, we see all the different parts we discussed in our last video. Again, we'll start with the nucleus and its 46 chromosomes. There is, of course, a 50% risk that the mutated X chromosome was inactivated as a bar body, but luckily for us, that's not the case here. Now, let's discuss mutations. DNA is different between different people. That's why we all look different. But some differences are well tolerated, like those between skin, hair and eye color. An enzyme, on the other hand, needs to function minimally well, or else the inner workings of a cell can get mixed up. That's why scientists define a reference sequence, so that deviations from this reference sequence can easily be detected. There are different kinds of deviations that show up in genes. First, we'll look at what can happen when a single DNA base pair is exchanged. For this scenario, there are three options. The best case is that the resulting codon codes for the same amino acid, a synonymous mutation. The structure of the protein is exactly the same, though there might be a slight effect on the overall fitness of a cell. Often, however, the result is an amino acid change. This is called a missense mutation. In some cases, the resulting strand of amino acids fails to fold, and no mature protein is produced. In cases where the protein can be folded, this mutant protein often has different properties, from barely active to slightly more active than the protein represented by the reference sequence. In addition to this, folding a mutant protein can be taxing on the endoplasmatic reticulum, leading to ER stress. In some other cases, such a mutation creates a new stop codon in the middle of the gene. Stop codons signal the end of RNA translation. The result is an incomplete chain of amino acids that usually cannot be folded into a functional protein. Those are nonsense mutations. Other types of mutations include insertions and deletions. An insertion or deletion of even a single base pair can cause a frame shift, meaning that all the subsequent codons can no longer be read correctly. These types of mutations often completely prevent the production of a protein. A special case are intronic mutations. Splice site mutations are easy to explain, as incorrect splicing means that part of an intron is included in the mature RNA strand, or part of an exon is missing from it. That's why splice sites are usually included in genetic testing. Deep intronic mutations are a difficult topic, so we'll discuss them in a separate video. For now, we'll just state: deep intronic GLA mutations can lead to an insufficient enzyme function and cause severe Fabry phenotypes. Genetic sequencing of the GLA gene often excludes the deep regions of the introns. If deep intronic mutations are known, physicians often don't treat the affected individual. Why? We wish we knew. A brief word on treatment options. In case no functioning enzyme can be produced, it has to be replaced by enzyme made in a laboratory. This is called enzyme replacement therapy, which is administered on a biweekly basis through infusion. If a mutation causes the resulting enzyme to be less active, there's the option to help the cell fold it better. This is what chaperone therapy does. Chaperone therapy is available as a capsule that is orally administered every second day. Fabry has a missense mutation causing an enzyme deficiency. The term classical Fabry disease is often used for cases with a near zero or zero enzymatic activity, typically involving severe kidney disease and associated with the male sex. However, classical Fabry disease should not be mistaken for typical Fabry disease. Over half the known Fabry mutations are missense mutations, and some of the more common missense mutations have a considerable residual activity. Because women have 2 X chromosomes, there's double the chance for a woman to inherit Fabry disease. Classical Fabry disease is clearly rare, but Fabry disease per se is much more common. So, Fabry represents a more typical case of Fabry disease. Now, after Fabry-N's GLA gene is copied, we have a near-perfect strand of RNA with only one small mistake. When this strand is translated, one wrong amino acid ends up in the resulting chain. The ER manages to fold a protein out of it, but that protein is less active than it should be. Additionally, it takes longer to fold, suggesting dysfunction of the endoplasmatic reticulum. Next, the deficient enzyme passes through the Golgi apparatus and arrives in the lysosome. Inside, different types of cellular waste are broken down. Here we will focus on the breakdown of sphingolipids, a family of lipids that form part of the cell membrane. The individual steps of the breakdown of sphingolipids each require a specific enzyme, and dysfunction of one enzyme stalls the breakdown process, leaving a specific waste product that cannot be broken down beyond that point. That sphingolipid part accumulates in the lysosomes, which is what is referred to by the term lysosomal storage disease. Each enzyme deficiency and associated waste product represents a different lysosomal storage disease. The sphingolipid part that accumulates in Fabry disease is abbreviated as GB3 or GL3. Lyso-GB3 or lyso-GL3 refers to a derived form. Further explanations on sphingolipids will be provided in a separate video. Now, we have lysosomal storage, but how does that cause organ damage? That's the part that's often conveniently left out. Fact is, we don't know. There is ongoing research into this, but much is still unclear. Here are 2 examples, one regarding the heart, one regarding the kidneys. GB3 storage by itself is not enough to explain the degree of cardiac hypertrophy, conduction abnormalities, and other cardiac manifestations. We detected complete resolution of GB3 accumulation in Fabry podocytes upon alpha-galactosidase A treatment. Despite robust GB3 clearance, dysregulation of the signaling pathways investigated was not reversed. Likewise, lyso-Gb3 is often used as a biomarker, meaning an increased lyso-Gb3 level in a patient's blood is used to determine whether this patient has Fabry disease or not. Still, quote, it has been reported in mouse models that lyso-Gb3 accumulates most in the liver and spleen, organs that are not affected in Fabry disease, end quote. Additionally, quote, plasma lyso-Gb3 levels fail to conclusively identify patients with milder phenotypes, particularly in females, end quote. We just don't know how lyso-Gb3 and disease progression are connected. So let's have another look at the table we presented at the beginning. Fabienne, please point out the first misleading bit of information. That's right, we have to correct the first line to missing or deficient enzyme. And what else? That might seem like a minor modification, but it's really important. We don't know how those deposits are related to the symptoms we see. On the other hand, with ER dysfunction, we have another possible cause of symptoms that's completely unrelated to lysosomal storage. Fabienne is quite convinced that endoplasmatic reticulum stress is more of a problem for her than lysosomal storage is, but her voice doesn't count. And in a paper published last year, yet another explanation for the broad range of Fabry symptoms was suggested. A number of genes involved in the immune system might become more active in Fabry disease. What does that mean? We are only just starting to understand what happens in Fabry disease. The question remains, why do physicians send patients home when Gb3 deposits cannot be detected, even though they have a GLA mutation and Fabry-like symptoms? What can I say? We wish we knew. So, Fabienne, that was a tough one. I think we did well. Thanks a lot. Now you can get your well-deserved rest. Our next video takes a special look at some common missense mutations that are sometimes considered benign. As far as we can tell, those conclusions rest on little circumstantial evidence and many assumptions. We hope you will be joining us again. Even if you don't, feel free to write and have a wonderful day.